Good day to you. Topic 3: Virus Quantification. This content is part of the module MBS 7117, Molecular Microbiotechnology. It's housed in the Department of Biomolecular Lab Technology and Molecular Biology, abbreviated as BLT, in the School of Biosecurity, Biotechnology and Lab Sciences, SBLS, College of Veterinary Medicine, Animal Resources and Biosecurity, COVAB, in Makerere University. I'm your facilitator for today's session, Dr. Penny Nansamba. The contents for topic 3 include the following. Number 1, definition and justification for virus quantification. Number 2, types of assays for quantification of viruses. Number 3, plague titration assays. Number 4, infectious dose 50. In section 1 of topic 3, which we are considering right now, we are going to dwell on definition and justification for the quantification of viruses and the types of assays we have available for the quantification of viruses. Learning objectives. By the end of this lesson, or topic 3, you'll be able to, number 1, justify virus quantification. Number 2, list methods for virus quantification. Number 3, explain plaque titration. Number 4, explain infectious dose 50. And again, in this section 1, we are going to mainly look at the justification and quantification of viruses and the methods used for virus quantification. Section 1 of topic 3: definition, justification for virus quantification, and the methods. Learning objective 1 and 2. By the end of this section, you'll be able to justify virus quantification and list methods for virus quantification. So let's start with the definition and justification for virus quantification. We start with the definition. What is virus quantification? It's the process of measuring the amount or concentration of virus particles in a given sample. Why do we do this? What's the justification? And number 1, it is to evaluate virus load in a sample so that we obtain an understanding of how much virus is present. Secondly, we want to assess virus infectivity and replication efficiency in experiments. Number 3, still on justifying why we have to quantify viruses. Number 3, to standardize virus concentrations for infection during experiments and vaccine trials so that we can ensure accuracy. And lastly, to track disease progression or treatment responses, for example, when we are monitoring the effectiveness of antiretroviral drugs. So let's look at various methods used to quantify viruses. And we are going to do a comparison of these methods or assays. So first we are going to look at the state or the character of the virus. Is it the whole virus particle? Is it infectious? Or is it just the nucleotide bits and pieces of the genome or the whole genome? That's what we mean by the character or state of the virus. Then we shall look at the principle underlying the assays, and we shall look at examples of such assays. So first, if we want to measure infectious virus particles. And the principle here is that we are only going to measure the particles that are infecting cells. We can use the plaque assays, which we are going to see in section 2, TCID50, which we are going to see in section 3, and the TCID50 includes the non-lytic viruses which don't form CPE, while our plaque assay is the gold standard. And the last assay which we have for this method is the focus forming assay, which is actually much more faster than the plaque assay. So our second kind of assay, or our second category of assay, are those that measure the total virus particles. They are not considering whether these total virus particles are infectious or whether they are non-infectious, but it is the whole virus particle with the genome and the capsid. So the principle underlying such assays is that they're usually antigen-based. Examples are hemagglutination assays or the ELISAs, enzyme-linked immunoabsorbent assays. And the advantage is that these are very simple and rapid. Our third category of assays are those that are nucleotide-based. So we need not have the whole genome and it need not be infectious. What we just need is part of the nucleotide. So the principle underlying this assay is that we're going to quantify the genomes regardless of their infectivity. Examples of assays in this category are the quantitative real-time assays, the PCR polymerase chain reaction assays. These are very sensitive. And now we even have the digital PCR, which are high precision assays. So let us reflect on what we have just studied using self-reflection questions 1. It's a multiple choice. And part 1 reads as follows. Which of the following methods measures only infectious virus particles? The key word is infectious virus particles. Is it the ELISA, quantitative PCR, plaque assays, or hemagglutination? Yes, it is the plaque assays. Question 2. Which is the main disadvantage of the quantitative PCR assays for virus quantification? A, it's time consuming. B, it doesn't distinguish between infectious and non-infectious virus particles. C, it's not specific. D, it cannot detect viral RNA. The key word here is main disadvantage of quantitative PCR. And you've got it right. It does not distinguish between infectious and non-infectious viruses. Part 3. The plaque assay is suitable for non-cytolytic viruses. True or false? So, does the plaque assay work for viruses that do not form CPE? False. We need to see CPE. Part 4. Focus forming assays require virus-specific antibodies. True or false? You've got it right. It is true. The last question. ELISA can detect virus proteins in a sample. True or false? True. ELISA can be antigen-based and viruses have got proteins that are antigens. So, in summary, the lessons we've learned so far, we've looked at the justification for measuring amount or concentration of viruses and we've looked at the different methods that can be used. The next step, refer to the lecture notes given to you on the study portal, the e-activities therein, and the textbook chapters or journal articles that we have recommended to you for further reading. Thank you.